Welcome, friends, to a podcast with interesting people. I'm Adrian Sinclair, and today my guest is Doctor from Rutgers University, Zhao, and he's in Department of Biochemistry and Microbiology. Here's a page from his Rutgers profile. He is a distinguished professor also at the Shanghai Jiaotong University, currently at Rutgers University, Department of Biochemistry and Microbiology, School of Environmental and Biological Sciences at Rutgers University. And we are going to be talking today about microbiome, health span versus lifespan in our series of podcasts where we talk about health, different diets, different things that impact our lives. I wanted to reach out to someone who deals not only with the sort of like what we eat, but how does that impact our system, our body, our microbiome. There's a lot of conversation about that topic. And so without further ado, I'd like to introduce you to Professor Zhao. Welcome to the show, Professor.
Yeah. Hi, everyone. Thank you, Adrian. And thank you for the opportunity to speak to your audience. I'm very happy to be here.
I'm very thrilled to have you. We were introduced by a mutual friend. And I was, when he said like, oh, you got to talk to Liping, because he's talking about microbiome. It's like people are looking into this right now. How is it impacting our lifestyle, but also lifespan? And you have a wonderful presentation at Rutgers where you talk about lifespan and lifestyle. Can you tell us a little bit more? What are the two? Just kind of tell us a little bit more about why are you talking about those two kind of juxtaposed together?
Yeah. So lifespan is the total length of time a person lives, and health span, another concept, is the length of time a person remains healthy. So basically, the ideal life would be your health span equals your lifespan.
Right.
So you live up to the last day and you stay healthy. That's the ideal human life.
Right.
But now we know almost, very few people can actually achieve that goal. Most of us would start to have a decline in our health, going down the hill and after mid-age. And eventually many people die prematurely and, from various chronic diseases, particularly in nowadays. So why our health span, for most people, health span is so much shorter than our lifespan? What makes us develop those chronic diseases, particularly, and die prematurely from our, potential biological age? So that is the fundamental question we would like to answer. But we are confident that one of the most important field, very exciting field, is the so-called human microbiome research that might bring some revolutionary concepts and technology, and for us to understand why our health span is so much shorter than our lifespan, and how we can do what we can do to to increase our health span and make it as long as our lifespan.
Right. So this brings us to the microbiome, your field of specialty. What is microbiome? If you can describe it to a layman, someone who has no idea what we're talking about, what is a microbiome?
So to understand what is a microbiome, then we need to understand what is a microbe or microorganism. So we know there are tiny, tiny organisms that we cannot see with our naked eyes. We must use microscope to see them, to study them. So all those tiny organisms, we study them in one field. It's called microbiology. And so among those so-called microorganisms, you have actually a very diverse range of life forms. You have fungi, you have bacteria, virus, those are people familiar, but you still have other parasites, single cell animal that we hardly can see with our naked eyes. But all these microorganisms, they actually we can find all kinds of microorganisms living in and on our body. So that's why the 1958 Nobel laureate, Dr. Joshua Lidberg, actually called human beings are superorganisms. Superorganism in his term means that our body is not just made of human cells. In addition, of course, the major thing is our human cells and make us human. But then on this human body, inside and outside, you can see trillions of hundreds of different species, trillions of cells of different organisms, living with us. So we call them combined human cells and microbial cells together. We are a superorganism. And all the microbes living in and us, we call them microbiome.
Mm. So I wanted to ask you, because I've heard it several times where people are saying that there's actually more cells or more bacteria and other things than our own human cells. Is that true, or is there more still cells of human cells and then some bacteria and all these other things that you mentioned?
Oh yeah, and at the beginning, so people have a rough estimation 10-1. That means 10 bacterial cells over 1 human cells. So in that sense, we are just 1/10 of human, because the whole body is made of 90% bacteria and only 10% human cells. But then some scientists really got serious about this, and they did a serious study. And from their research, they say, okay, it's actually about 1-1.
Okay.
And
that makes me feel better.
I would say it ranges from 1-1 to 1-10. And because there is a trend that when we have a health problem, we often have overgrowth of bacteria in our gut. So in that case, you would have maybe increased the proportion of bacteria over human cells. But roughly, normally we have a 1-1 ratio, in terms of human cells and microbial cells. Even 1-1 is still a lot, right? That means you are only half human.
I was going to say, it makes me feel like, only half of me is me and the rest is just some additional,
that's your better half. That's
right. That's my better half.
That's the other half. Biological better half.
Biological better half. So one of the questions that people will probably have is, why is this microbiome so important, right? We're living in this balance with these bacteria and viruses and parasites and all of these different things living inside of us. Why is it important to keep that in balance, and how do we even do that?
Okay. Well, this is actually the most fundamental question scientists in the microbiome field try to answer. In general, we are interested in microbiome because they live in and on us, and we need to realize that for the whole human microbiome, 90% or even a higher proportion actually all lives in our gut. So our gut is a place where most of our microbiome stays and lives. And so all kinds of bacteria, they live there and they grow on a daily basis, because every day they grow, and then part of them will be removed together with bowel movement. And so they need to multiply and make that compensate that loss so that they can stay in the gut on a relatively stable level. So because they are growing all the time, so at least some of them can produce some metabolic waste. So when they grow, they take nutrients and then they dump waste outside of their cells. And many of those metabolic side products, they are biologically active. So that means some are beneficial. For example, a group of compounds called short-chain fatty acids, and they are beneficial in many ways. So we can come back on that later. And then, of course, some are producing toxins, some are producing antigens, can stimulate our immune system, induce inflammation. Others can produce neurotoxin, and some produce carcinogens, which may induce cancer. So all kinds of toxins and detrimental compounds may be produced by some, so-called bad bacteria in our gut. So that's why we need to understand, first, are there any functions which are essential to keep us healthy, and they can only be provided by the gut bacteria? So that means gut bacteria becomes an essential partner for keeping us healthy. And yes, so more and more research, building evidence, and to show that, for example, what we call the short-chain fatty acid producing bacteria, and particularly acetic acid producers and the butyric acid producers. So they provide those short-chain fatty acids, and those short-chain fatty acids, they provide energy to our gut cells. On our gut surface, there's a so-called epithelium cells, and that's the outer layer of our gut. Those cells, they grow very quickly. Every 3 days, the old cells will come off, replaced by new cells. So they need a lot of energy and other nutrients. The major form of energy they need is butyrate, locally produced by some gut bacteria. So this is a very interesting example where you grow bacteria, and then the bacteria keeps you healthy. And there are also other examples of bad bacteria. We need to make sure that they are at a very low level so that they don't produce too much harmful products. So it's a fundamentally important question. We need to understand what kind of bacteria they are beneficial and protective to our health, and what kind of bacteria are actually pathogenic and infectious and detrimental and toxin producers. And there are still others. They may be just the so-called commensal. They are neutral. They just stay there, take advantage of the energy nutrient-rich environment, but they either do no harm, they all do nothing substantially good.
So
we need to understand that. Yeah.
This is very interesting and complex. Fascinating that someone can actually discern what the heck is going on in the gut, because it's like such a rich and diverse environment. And my question is, are there bacteria that may be good that may become pathogenic if they overgrow, or do you have, because in your presentation you talk about guilds of bacteria, like they form these relationships and they form these groups and they interact, and is it the case that we have bacteria that we just know are beneficial to us, and then bacteria that we know are bad for us, or is it not as simple as that? And like bad bacteria may become good, and good bacteria, if overgrown, may become bad. How can you make those distinctions?
Yeah, well, you just mentioned this is such a complex system and also dynamic, changing almost all the time.
Right.
So how can you tell which is good, which is bad, and
right.
Or sometimes good, sometimes bad? How can you just even study? People just cannot imagine. When you have thousands of different kinds of bacteria, and with such a huge population density, so how can you just dissect such a complex system? Well, I think that this is need to thanks to two fundamental developments in technology. And one is the so-called next generation sequencing technology. So now we can sequence a DNA sample at a very, very high throughput. For example, now we can sequence DNA we can generate 8 million reads. Each read is a short fragment of DNA, like 150 base pair, just like 150 letters. But you can generate 80 million in one go, and for one DNA sample. And then you generate so many small pieces of DNA, and then you can just piece them together and then you can recognize bacterial genes. So in this way, we don't need to isolate and culture bacteria from our gut samples. We only need to extract all the DNA and have a massive sequencing of the DNA so that you generate a huge amount of data from each human gut sample. Usually it takes sometimes you can actually go inside the gut and take sample from some location. But for most of the human research, you can just use a fecal sample because half of the fecal matter actually bacterial cells. So that's why you can just use that to detect what kind of bacteria once live in the gut. And they come out, and so you analyze their composition change. So we need to thanks to this next generation sequencing technology, which give us the capacity to sequence almost everything in the gut. We know that DNA is fundamental to life. To recognize a bacteria, understand what it can do, is if you can sequence its genome, can get all the genomic sequence of bacteria, you pretty much have confidence in how to understand their function and behavior. So this next generation sequencing technology paves the way for us to recognize and characterize each major bacteria in our gut. And also in samples where they change over time or in responding to our dietary change, when we are developing the disease progress from pre-disease, a mild form of the disease to severe form of disease. We know some bacteria would be increased, some decreased, some not changed. When you change your diet, some bacteria would be promoted, others would be actually reduced, suppressed, and most of them probably may remain unchanged. So there are so many things can diet and health, disease progression, and drugs can change the bacteria and they would respond. How can you detect their changes, quantify their changes so that you can analyze them? So this next generation sequencing technology make us can go down to DNA level so that we can get a very good understanding on those. So this is one reason. The other reason, equally important, is the powerful computational technology now we have. So you can generate such a huge amount of DNA, 10 gigabase pair per sample, and then 100 samples, you get 1 terabyte, and easily it's a real big data.
Right.
So you need a place to store it. Then you need enough computational power to understand it, to mine the pattern. So these two technology actually give us the capacity to analyze variations of a microbiome along with variations of our health and disease and change of our diet and treatment, so that we can correlate changes in the microbiome with the changes of our health. And then eventually we can identify those members which are increased when we are getting disease. And so they show a positive correlation with disease.
I see.
They are potentially detrimental or pathogenic. And then we have other members which are negatively associated with disease. Then they are potentially beneficial, protective. So that first you identify all the related or correlated members of the microbiota with our health or disease.
Makes sense.
And then now you know their DNA, you know who they are. So you try to isolate them from the gut microbiota. So you separate them from all the rest of the other irrelevant bacteria. So you focus on either one single bacteria or a group of bacteria, and they stay together. They cannot be separated, but you can isolate them as a consortium, as a group. So after you isolate the live bacteria, either as a single member of the gut microbiota or as a consortium, and then you can put them into animal model. You can create the so-called germ-free animals. So where you put your candidate bacteria identified and isolated from human, and then put them into germ-free mouse gut or germ-free piglet's gut, or even germ-free non-human primates gut. And so they can colonize the gut. And then you can see whether you give a disease-triggering condition to this animal model, animal containing human bacteria, you give the disease-triggering condition, can you reproduce the disease? Or at least some of the phenotypes? If yes, now you have a new human disease model, right? And then you can use all the currently available very powerful molecular tools, try to understand what kind of effector molecule produced by colonizing bacteria and which bind to what kind of receptor on the cell surface of animal gut, and then triggering what kind of signaling transduction pathway, and then eventually change the expression of what genes, shut down what kind of genes and turn off what kind of genes. So all these, signal transduction cascade pathway eventually change human disease phenotype. So this is a daunting task, but it can be done now. All the pieces are now in place. So we have pretty much the technology to work on dissect this complex system.
That is so fascinating, and thank you for explaining this in such a way that even I can understand. It was it made sense how this whole progression of scientific experiments and the process, and I'm sure like the whole maybe machine learning or even artificial intelligence right now that you guys probably employ in sifting through these terabytes and terabytes of data help you identify these groups of bacteria and how they behave. But it makes sense to kind of put those in the animal analogs and how you can kind of then introduce external stimuli and see how they respond to kind of verify if they are good or bad. So I get that. That is just so smart. And thank goodness we have people like you who are studying this, because this is a very complex field. And I was just scratching my head like, how are you making sense of anything that's going on in this environment? Because it is, like you said, so dynamic and just such a huge variety of different organisms. So it's like you don't know how to figure what's what. So now bringing this to everyday person's life, how can people, because you mentioned that diet obviously affects the microbiome, right? What we eat. Sometimes when people are sick and they need to take antibiotics to deal with some kind of infection, that affects the microbiome. And what are your thoughts on how people can provide their own bodies the best possible tools, food, and things in order to stimulate, to grow this well-balanced and healthy environment?
Yeah, well, everybody almost, understand the principle that our diet is fundamentally important to our health. So there's the concept of good diet, bad diet, all kinds of, dietary patterns have been proposed, and this is really a very important field. Excuse me. However, we know actually our diet impact our health by two ways. One way is of course directly feeding our own human cells. So our diet provides the nutrients we need for our body to grow properly and function properly. So that's the direct contribution of our diet. But there's another missing part people ignored for too long. But now from microbiome research, we understand our diet actually can also change the bacteria in our gut. So change their composition and change their activity, change what kind of bioactive compounds they can produce. And then in return, those change the microbiota would impact our health. For example, if your dietary pattern, instead of promoting and maintaining the essential healthy bacteria, but actually reduce the healthy bacteria, promoting some kind of pathogenic toxin-producing bacteria, then eventually the whole gut microbiota becomes a disease-causing agent. Instead of a partner for health, it becomes a pathogen for disease, because overall, the gut microbiota start to produce something which damage your health. For example, if some bacteria overgrow in our gut, and they can produce some antigens which can overstimulate our immune system, so they can induce inflammation. If they induce inflammation throughout our body, eventually we have, this is my own field, eventually the long-term systemic inflammation in our body eventually will damage insulin receptor, increase insulin resistance, and induce obesity and type 2 diabetes, and then they may also affect our neurons and affect other functional cells, so they can contribute to development of complications. And some other bacteria we also mentioned, they can produce carcinogens, so that they can promote cancer development. So all this depends on what kind of nutrition they can get. So if they get their own favored nutrition, they can grow and they outcompete other bacteria, and then they become a major member. So now their bioactive compounds would become a major player in affecting our health. And so that's why when we look at our diet, we need to realize that our diet should include two kinds of nutrition. One for our human cells, and the other should be satisfying the needs of beneficial bacteria. Mm-hmm. So this is a feed my microbiome dietary scheme. Otherwise, if you take extremely good care of the nutritional needs of a human, you ignore the nutritional needs of a healthy microbiome, then eventually your microbiome would be overgrown and dominated by potentially pathogenic and disease-causing organisms. And then they would produce detrimental compounds and other virulent factors, and then eventually either induce disease or aggravate an existing disease. So this is why diet is so important. And if you look at our gut, it's just like a bioreactor, like a fermenter. And it's a long tubing system. And so almost every part, particularly the lower part, highly populated by bacteria. They need nutrients to grow, to compensate the loss, on a daily basis, so they can maintain a relatively stable population. Otherwise, after a few days, they would be washed out, completely. And so anything in our diet which are either non-digestible or undigested would become available to our gut bacteria. So that's how gut bacteria can get nutrients from our diet. So molecules which we cannot digest. And the major example is dietary fibers. So they are complex carbohydrates. We don't have enzymes to break down those carbohydrates, so we cannot use them. But they can be used as energy source for many potentially beneficial bacteria. And this is actually why dietary fibers, particularly those which are fermentable dietary fibers, actually beneficial to human health by way of promoting healthy bacteria.
What are some of the examples? What are some of the examples of dietary fiber which is fermentable?
Yeah, this is actually a question pertaining to everybody's daily life. When you talk about having more fiber, people in general understand that have more fiber is good to your health, right? But you probably would hear the advice that when they say you need to take more fiber, then automatically you go to the market, grocery shops, and buy more fruits and vegetables. But unfortunately, that is wrong, because dietary fibers in fruits and vegetables are mostly non-fermentable. So that means actually using vegetable and fruits to increase as the major source of your dietary fiber, you have two problems. One problem is the density of dietary fiber is actually very low because most of the part is water, right? you take a very bulky meal with vegetable, you actually end up with not much dietary fibers because most of the fresh vegetable, they are just
A lot of
water. And that's the second is the form of dietary fibers in vegetables and fruits. They are non-fermentable by most of our gut bacteria. So they just pass through. Of course, they are beneficial. They can help you, increase the bulk of the meal, and it can help the transit and the motility of your gut. So it's helpful. But in terms of feeding the good bacteria, they cannot do much.
Right.
So, but
what is the answer? What are the fermentable fibers? Give us the secret.
So I think it's just people just a fiber is a fiber, so don't care whether it's fermentable or not. So then you need to really to look at so-called adding fermentable fiber, and then so most of the fiber in grains, in plant seeds, they are fermentable, and like resistant starch and other cell wall components of plants of the plants, they are so-called they are fermentable, mostly fermentable. There are also other manufactured dietary fibers, such as the so-called prebiotics. Mm-hmm. Most of them are highly soluble because they are short-chain oligosaccharides. The structure is very simple compared with natural fiber. So they are highly soluble and easily broken down. So they are highly fermentable. Mm-hmm. But because they are highly fermentable, and because it's a form of energy, fuel for gut bacteria, and because it's highly fermentable, it's actually accessible to not just the beneficial bacteria. Even many not so healthy bacteria can also feed on those easily fermentable. So it's not a simple question. Mm-hmm. You need to, and also there are, if you take too much fiber, there are adverse gut reactions, bloating, constipation, diarrhea. And all those reactions is actually a reflection of both the so-called dysbiosis of our gut microbiota and also the wrong combination and amount of the dietary fiber you feed them. For example, many people already have a form of dysbiosis of gut microbiota called small bowel bacterial growth, overgrowth. So basically, it's we have some bacteria in our small intestine, but we should not have too much, too many and too high population level. But many people actually have a lot of bacteria overgrowing in the small intestine, so they can feed on fiber and they can produce gas. So when you take a high fiber, easily fermentable, high fiber diet, within a short period, within 1 hour also, you start to feel bloating. Because those bacteria in your small intestine start to feed on the fiber, grow, but also produce a lot of gas. So gas built up in your small intestine cannot go up, cannot go down easily. So you start to build up in the middle, and then you feel really pain, and sometimes can be life-threatening if you have ulcer or other gut conditions. Mm-hmm. So it's really not just a simple question, just eat more vegetables, more fruits, you'll be fine.
Right.
That's not,
I know.
So
somehow I knew it's going to be more complex answer. Somehow I knew it's going to be a complex answer, and because it is a complex problem. So without the benefit of technology that you have at the university, where you can study things, pull out data, and sequence them, and all of that, how does a regular person, manage this very complex environment? I suspect that one of the answers is going to be, well, how do you feel when you eat? Right? That's kind of like your feedback. Do you have diarrhea? Do you have constipation? Like those are kind of like the some of the things that you kind of can figure out what's happening in your body. Bloating, obviously. So those are some indicators. But what else can people actually do? It sounds like everything is kind of individualized in a sense that everybody has to kind of figure out how their system is working. So maybe you need to add a little bit of these probiotics, see how your body reacts. Maybe if you have too much, then, kind of like fine tuning everything. But what have you found, as a approach to kind of figure out like how to properly maintain your microbiome?
Yeah, I like your question, because this question is not for patients. It's for healthy people to maintain your health, to maintain your healthy microbiome. If people, if you are a patient, I don't recommend you, you really need to see advice from your doctor. But for healthy people, to maintain or to achieve an even healthier status, we know health has a very wide range, right? And so you really, when we look at the professional in the so-called healthy human's gut, most people actually have maybe 2 or 3%. But with a proper microbiome nutrition, diet, with a diet included the proper microbiome nutrition, you can increase that at least by tenfold. You can increase that to 20%, 30%. Imagine if you can do that, and then a major part of your microbiome would become beneficial, and they can just provide the necessary beneficial compounds to you and to suppress pathogens and keep you healthy. But as you mentioned, regular people still don't have access to the just emerging technology to analyze the microbiome and do some tests,
right,
identify exactly what's wrong or what
right,
need to be promoted, and have a personalized nutritional pattern for you. All this, it's coming. It will become a leading form of technology which can affect everybody's daily life. It's coming, but it's still not available to everybody. So what you can do in your daily life and try to maintain a healthy microbiome, for doing anything, we need data or we need measurements, right? But if you don't have those highly advanced analytical technology, how can you monitor your health?
Right.
Yeah, well, there are many things you can do. And first, you need to look signs that indicate potential problem. As you just mentioned, if you have irregular bowel movement, either constipation or diarrhea, and other gastrointestinal symptoms like bloating, those are for sure signs that your gut microbiota is not well. So if they are serious enough, then of course you need to seek help from doctors. But if they are mild, just some indication that some problem might be there, and then you need to correct your diet and try your diet. But there's actually very, I know in Western culture, people don't talk about bowel movement a lot, right, in the public.
I'm changing that every podcast. I'm talking about poop.
but actually so if what would be the best indication of a properly functioning microbiome? It's actually look at your poop and golden color, odorless. No strange odors, not smelly. And color is almost like a yellow or golden color, and also cylinder form. Mm-hmm. Okay? Those three things, people can try. Not many people can achieve that. But if you can properly manage your diet and manage your lifestyle, and then your microbiome will also respond. And eventually you would have, of course, regularity, and a regular either 1 or 2 times a day. And also the poop should be yellow or golden color and cylinder form. and also without any, not smelly, odorless.
Wow, that's
So those, for example, there is a very special odor. It's actually from a chemical called indole. So indole is a side product produced by some bacteria when they use the amino acid tryptophan. So if people have an unbalanced diet, too little fiber, too much protein, then you end up with a lot of protein not digested and absorbed by human. So you leave this undigested protein to gut bacteria, and then you create an unbalanced nutritional environment for gut bacteria. So many bacteria which rely on protein to grow, and they can grow. When they grow, they produce toxin. Indole is one of them. So indole is the special odor coming from poop. And that's why, and also another toxic compound produced by bacteria when we have an imbalanced diet, have a bad microbiome, is the so-called hydrogen sulfide. We know that's the smelly gas produced when the egg rotten, right?
Right.
That's the rotten egg smell. And so even if you can reduce the production of indole and also production of hydrogen sulfide, that's an indication of a healthy microbiome. But overproduction of those smelly and compounds is actually indication of too much toxic compounds produced by the gut microbiota. So that's why I say, and also why is the yellow color? Why yellow color?
Right.
It's just because our bile is yellow. So if the many bacteria can actually work on the bile acid and transform them from primary bile acid into secondary bile acid, many of them are actually carcinogenic. And also they are, detrimental to our health, maybe also in other ways. So when they become, when the yellow color, the primary bile acid transformed into secondary, and then they lose the color and they become dark brown or other more dark colored, more smelly and toxic compound.
Right.
So that's why I say, if you don't have any highly advanced equipment or access to DNA sequencing, microbiome analysis, and the sure indication of a healthy microbiome is look at your poop every day.
So the reason why I was kind of smiling as you were saying this, it just reminded me of, watch animals, what animals do. Like if you have a dog poops, they will turn around, they will smell their own poop. And that makes me think that maybe they're just diagnosing like, hey, am I healthy? Is what's going on over there? So maybe there's something to be learned from the animals in that respect, because watch your poop, and if it's too smelly, maybe there's some kind of issue in your gut. So I'm glad that we have this very sophisticated device over here that may give you some inklings of what's happening in your gut. One of the things that I want to pull up this website of Rutgers website that we're looking at in your research interest, and I wanted just to tie into what we were just talking about. One of your bullets over here is formulating interventions, integrating traditional Chinese medicine and medicinal foods into modern day diet to modulate the relationship between gut microbiota and humans for achieving preventative healthcare. And my question was, Chinese medicine has such a rich history of herbs, herbal and different compounds, found in nature to kind of heal people. And I'm curious, what have you, in your research at Rutgers and in your whole career, what have you implemented from the traditional Chinese culture and knowledge in this respect into this field? So it's kind of marrying the traditional with the technological right now. Have you had any chances to kind of incorporate and see if any of those traditional things truly have impact? And what were some of your findings?
Yes, we actually already published quite a few high-impact papers by integrating concepts and materials, ingredients from traditional medicine and into modern-day diet, and to change the microbiome, improve health, particularly in obesity and type 2 diabetes. and it has potential to other, health problem as well. So more specifically, there is a written nearly 5,000 years of written medical record in China. So traditional doctors, they need to be able to write, those are scholars. And so there's a tradition that whenever they see a patient, they see a case, and they help somebody, they will take a very detailed record. And so almost every well-known traditional doctors throughout the history, they left, a whole bunch of books and monographs and medical records. And so there's a rich, some of them have lost, but many actually kept until today. So that's a very rich resource to look into. And we did two kinds of research, and one is we take advantage of a group of plants. It's called traditional Chinese medicinal foods. It's an officially published list of plants by China Ministry of Health. And so plants in that list, they are regulated by Chinese FDA both as food and as medicine, because they have been used as medicine, as drugs in Chinese medicine for over 1,000 years. So that because they are so safe and you can take them, integrate in your daily life, so eventually they even become part of our menu. And so then eventually the Ministry of Health, they publish a list to say that, okay, if you use plants on that list, either they are fruits, seeds, or leaves, and then they can be regulated as food and not as drug. So we take advantage of that. We integrate whole grains, traditional Chinese medicinal food, and the prebiotics all together, and we developed a new dietary scheme and try to change the microbiome. But if you look at the traditional medicinal foods, they are rich in two kinds of nutrients. One is the so-called polysaccharides, and they are actually, not surprisingly, non-digestible carbohydrates. So if you like, you can call them dietary fibers. But they are usually not part of our diet because they are part of the medicine. But in China, you can use that as a diet. So they are highly rich in dietary fibers, and technically. And so this is one major component. And the other is they have various phytochemicals, those colored compounds, brown colored, yellow colored, green colored. And those are polyphenols, alkaloids, and isoflavones. So all those small molecule plant metabolites, plant phytochemicals, and many of them also have very potent activity on changing the microbiome. And for the past 100 years also, when Western medicine came into China, that generated a long-term dispute among Chinese society. Some say, oh, we should combine Chinese medicine with Western medicine. The others say, no, Chinese medicine is not science. There's no, cannot understand why it works. So we should just stick with the new Western medicine and give up the traditional medicine. So there's always, even today, a debate on that. Yeah, but now one major reason people cannot understand Chinese medicine is if you look at the decoctions, cooked from various herbs, milky, black decoction, very bitter very often. There are many phytochemicals. Most of them are so-called, have very low bioavailability. So that means we cannot absorb them. They don't go into our bloodstream. We know that in Western medicine, if a drug cannot get into bloodstream, it cannot become a drug because the target is human cells. To reach human cells, you must be highly absorbable. A good drug should have a high bioavailability, right? But if the ingredients in your medicine are hardly absorbable, cannot get into the bloodstream, how can it work? So that's why people just do not, but now we know they don't need to go into our bloodstream because they stay in the gut. They still suppress pathogenic bacteria, but promote the beneficial ones. When they do that, they change the balance of our gut microbiome. So they promote more beneficial compound producers, and they reduce toxin producers, and that actually improve the symptom. And so you don't need to go into the bloodstream if your target is the gut microbiota.
Right.
So then it makes total sense, right? So we have papers published to show, some pure compound or mixture of mixed decoction from several herbs. They change the microbiome. They actually improve diabetes or other metabolic disease, and of course, they also have potential to other diseases. So it's and then for the polysaccharides, for the dietary fiber part, and they promote beneficial bacteria, and so that's why I think microbiome research. I call it, it has several functions. First, it's a window. So that means look at what's going on in your gut microbiome, particularly, what's in there and what kind of bacteria are overgrowing, whether they are beneficial or detrimental, what they can produce, and what kind of compounds they produce can really get into our bloodstream and affect our health positively or negatively. So looking at the variations of the gut microbiome, it's actually a window of our health.
Right.
Because they respond to, and then it's also a bridge. Just because now suddenly, no matter what kind of knowledge, whether it's coming from Western modern medicine, Western food and nutritional science, or traditional medicine in other developing countries, no matter what you have done traditionally or in modern days, and to understand human health, all those knowledge now, maybe not all, but many of those knowledges can now be bridged. Just looking at, okay, you have a traditional way of improving human health, fine. Herbal medicine, acupuncture, massage, whatever you do, okay, let's see how the patient's microbiome respond to that. We have a sequencing and a computational platform to monitor even a very small variation, among thousands and millions of variables. And then if you have a modern medicine, a wonder drug, fine, give it to your patient. And we don't interfere with your treatment. Do whatever you want, but we look at how your patient microbiome respond. If your drug or treatment or whatever you do promote the beneficial ones, then it's a good treatment. But if it promote the pathogen producing and the pro-inflammatory bacteria, even though in the short term it may help alleviate the symptoms, but in long term, it definitely would damage the long-term health of your patient. So now you have such a powerful tool to use microbiome as a window for monitoring health, but you're also using microbiome as a bridge to integrate all human knowledge on health and the disease and any available intervention we use to improve our health.
I always kind of cling to the the phrase that I like, which is like the truth is somewhere in the middle, and it seems that what you're describing over here is a fine dance. It's a fine balance between different things that impact our systems, because our bodies are just so complex that there's a lot of moving parts. There's a lot of systems and subsystems. We need to try a lot of different things and not be very definitive about like, oh, this is this drug is doing this and it's great at it, and it's so therefore it's the best. But there's also like the side effects. There's the impact on gut microbiome, and that has to be looked at as well. And so I'm glad that you guys are developing technology that allows collection of that data and processing to kind of try to unravel that puzzle. And there's something to be said about the long, history of medicine from China and other places where people have experimented with different plants and herbs, because those are chemicals. I mean, we think of like plants as like, oh, okay, well, I can eat some lettuce, but then, other things that like these plants are very good chemists. They can produce very complex compounds that can impact us in very unusual ways sometimes. And so it is a very, very complex field. So I'm happy to see that there is some kind of convergence between science and like these traditionally, derived sources of knowledge, because they were developed over, hundreds and hundreds, if not thousands of years, where people were trying different things. And I'm kind of glad that you have this balanced answer and not like this is right, this is wrong, and it's just one or the other. The truth is in the middle, isn't it?
Yes, the middle way.
The middle way, exactly. The way of life.
Yeah, Confucius concept.
Confucius, exactly.
Yeah.
So let me ask you a question about, so you mentioned how that this is coming in the future, these diagnostic tools where someone is going to be able to either go to a doctor and get these either lab tests or go to some kind of company and get, provide a sample, and that sample is going to provide a very individualized diagnostic tool, a report that will let them know, hey, this is what's happening in your gut, and here's what we propose you do, instead of just looking at your poop and smelling it. And how far do you think that is away from us? Is it like a year, 2, 5 years? What do you think is going to happen in that space?
To be on the safe side, maybe 5 years. And even though now you already have, many companies claim that they can analyze your microbiome and provide some advice, what you eat or help you diagnose the problem. But I would say the science is still a little bit immature. And we still need accumulated data, for example. And there are also protocols which are still evolving. And for example, we still don't have the healthy reference of the microbiome. So that means we know everybody has a different microbiome. And even I'm healthy, but during a year of time, if you look at my microbiome on a daily basis, of course, you will see changes from day to day.
Right.
But I'm pretty much healthy throughout the year, right? Like 18-year-old healthy boy, healthy girl, within the same year, you hardly see any major changes in their health conditions. But if you look at their microbiome, it may change. There are still changes, fluctuations, and also that means there's a so-called normal variations from person to person or in the same person from day to day. Those variations, obviously, we can tolerate, right? Some bacteria can increase today, almost disappear the next day, after 3 days, go up again. So you see some bacteria, they change dramatically when we are at the so-called steady state, when we are at the healthy state. That means those variations we can tolerate. They don't affect our health. And so those are the so-called normal range of variations. And of course, there are other variations that may probably are problematic, may drive a disease or aggravate a disease. So right now, all these normal variations and abnormal variations, they are mixed together in such a complex system. So we need to dissect that.
Right.
And we still need to build a reference database, at least, for example, now I'm part of the International Human Microbiome Consortium, and we are trying to promote a project where we want to sequence 1 million healthy people throughout the world, their microbiome. So that means if we can do that, so I think the beauty of this project is we should use the same protocol, using the same technology, same method, same protocol, standard control, quality control. All this should be the same. And then we sequence people from all over the world, and then we put the data in the same database. If we can achieve this goal, we probably would have a very good healthy reference database for identifying, telling the difference from healthy and the disease.
Right.
And but this currently it's a challenge because every nation, even labs in the same country, they have their own protocol, their favored protocol to do things. Mm-hmm. And so if you don't have a same protocol to sequence this 1 million, that makes no sense because
you're going to have different data.
You cannot combine your data. You cannot compare your notebook. But anyway, this kind of effort is needed. Mm-hmm. And before we can really do large-scale, day-to-day microbiome analysis and use that as a reliable marker to help us manage our health.
Right.
And we still need, to do that. So that's why I say probably we need at least 5 years.
5 years. Mm-hmm. Yes. I've heard of some of the companies that you mentioned in terms of like they do like a DNA testing on you to kind of help figure out like what kind of foods you specifically may be either allergic to or like have bad reactions, or maybe some foods that are good for you, specifically good for you. It just adds on to the complexity. One of the questions after watching your presentation at Rutgers was, we have different cultures, different places in the world that have different diets, right? I mean, the Inuit in North Pole or like in that Arctic Circle will have different diets. They will have high fat, high meat, high protein, and very few vegetables and fruits and plants in their diet. And then you have other other groups of people living in other places which are like highly focused on fruits and vegetables and not a lot of meat. So how do you combine all of those different, even regional varieties? And you can argue that, there's healthy people here and there's healthy people over there. That just adds on to the complexity of this big puzzle, because then you can't just prescribe this one solution like, oh, you should eat this and this is good for everyone, right? So how does that affect what you guys are researching? Are you looking into different groups and their microbiome? So that just adds on to the complexity.
Yeah. So like the 1 million healthy microbiome project I mentioned, the idea is exactly that. Different ethnicity, different geographical origin, different traditional dietary patterns, even different gender. All these things are the major confounding factors for affecting the composition and activity of gut microbiome. And so traditionally, people don't move around a lot. One person would stay in the village throughout his life and seldomly to travel to other places. But now we are moving around and we move away from our hometown, and we overnight we change our environment and our diet. So our microbiome actually, has experienced a lot of challenges, a lot of, medications, antibiotics, which can change them, and diet changed abruptly. So then over the, millions of years, we actually stay in a place and we live on the traditional diet with local food. So our microbiome actually adapt to our local diet. So the composition of our local healthy food can support a local healthy microbiome. And that, becomes a normal pattern for local people. But when you change your diet, abruptly, for whatever reason, then your microbiome will change. And but that change, for most of cases, is actually changing to the unhealthy direction, because originally those bacteria which are promoted and dominate your microbiome and supported, sustained by your local diet, they are the ones which can keep you healthy, provide the essential functions and suppressing pathogens. But if you don't feed them, if you don't provide the energy source they need, they would decline. And then they no longer can suppress other unwanted pathogenic bacteria. So then other pathogenic bacteria would start to overgrow. So this is why, for the early days when Chinese scholars and students moving to US to study, in the first 10 years also, they don't develop the Western disease. But after that, you start to see asthma, type 2 diabetes, IBD, all kinds of typical American disease start to also develop in these immigrants, new immigrants here. But if you compare their home, their brothers and siblings and families in China, they still stay rather healthy. But then when China developed its economy and the people start to adopt a Western lifestyle and Western dietary patterns, boom, you have type 2 diabetes, obesity, all the Western disease. Now it's all over the place. And so it's, I think your healthy microbiome requires a carefully structured, healthy diet to maintain. For example, we have a successful clinical trial which was published in Science 2 years ago. And we provide a high fiber diet to type 2 diabetes patients and improved this group of beneficial bacteria. And for 3 months, and we see reduction of HbA1c, fasting glucose level, and they lost some body weight. So you see significant improvement of their health conditions. And but then we stop the trial, and so they go back to their normal diet. And then after a year, we get them back. So if you look at the baseline, which is a disease state, and then 3 months after the intervention, which is much healthier microbiome and healthier, body. But then after another year, when you get them back, and they have the old microbiome back, and they had the worse health. And so the beneficial bacteria, they need to be maintained by a healthy diet.
Right.
If you don't provide that diet, they would decline. When they decline, you don't see what kind of pathogens and toxin producer would overgrow. It's sporadic for different people. And this person may have a carcinogen producing bacteria overgrow. That person may have neurotoxin producing bacteria overgrow. Others may have endotoxin producing bacteria overgrow. And then they develop cancer. They develop Alzheimer or other neurodegenerative disease, and some develop type 2 diabetes. But then it looks at the surface, different pathogens drive different disease, just like infectious disease. However, the root cause is not overgrowth of the pathogen, it's the loss of the protective and beneficial bacteria, and then allow overgrowth of different pathogens. So that you need to go back to rebuild a healthy microbiome dominated by this beneficial bacteria, and then they would automatically, ecologically suppress those pathogens.
Right.
And so that's the probably eventually the ecological solution to the health problems.
So let me ask you a question. I have a couple of questions just queued up in my mind over here right now. One of them is the effect of gut for on your brain. That connection between gut and brain, affecting neurons, affecting your brain development. Obviously, one is feeding the other. Like your gut is absorbing the nutrients and it goes into your body, your system, and then gets into your brain to feed that as well. What are your thoughts on an opinion on the connection between gut and brain? Healthy gut, healthy brain, or no?
Well, I would say yes. And this is not my particular field. We have our own lab only have a small piece of data to show that. That is when we feed type 2 diabetes patients with a high fermentable fiber diet, and we promote this group of acetic acid and butyric acid producing bacteria. And then we see a gut peptide hormone called PYY, increased. So this hormone is actually, so the mechanism is you feed the fermentable fiber to the gut microbiota, then you promote bacteria, and then they produce acetic acid, butyric acid. And these bacteria produce the short-chain fatty acids come to combine to a receptor on the surface of our gut epithelium. And then this cell is called L cell. So this L cell, when it's stimulated by the acetic acid, butyric acid, they will produce many hormones. One hormone is called PYY. So PYY can actually migrate to our brain and increase our satiety. So even though you have a low-calorie diet, you don't feel that hungry. Because
interesting.
This is So this is one example how gut bacteria eventually change our food behavior. And we have data to show that even a special group of patients, a genetic form of children called Prader-Willi syndrome, they have a very severe form of hypophagia. They seek for food all the time, hungry all the time. But when you put them on this microbiome diet, after a few weeks, you see significant improvement of their food behavior because they feel satisfied. And what's very interesting is many parents told us the way they change their food behavior is before they were on the intervention, when they found some food, they immediately put into their mouth. And they eat immediately. But after a few weeks on the intervention, when they found food, they don't eat them, but they hide them somewhere. They keep it somewhere, and so psychologically, they're still crave for food. But physiologically, they no longer need the food because you feed them properly, so they feel satisfied. But psychologically, they still have that kind of addiction to the food. So it's really there are other examples. Now we know almost all the major neurotransmitters, like serotonin, other, can be produced by some gut bacteria. And also, almost every major neurotransmitter can be degraded as nutrient by some other gut bacteria. So why bacteria do that, right? Imagine if they can produce some neurotoxin, neurotransmitters, or reduce the concentration of other neurotransmitters, that probably eventually would affect our thinking, our behavior, our psychology. So, depression, for example, if you transplant the total gut microbiota from depressed patient to germ-free mice, those recipient mice, they become depressed.
That's interesting.
So that means this disease can be transferred along with the microbiome. You get rid of the original genetic background, the original dietary pattern. Everything is no longer human, but only the microbiome is human. You put that into germ-free mice. Within a few weeks, you see the mice develop depression. And so that means, so there are other examples. So basically, well, this is for disease, but can the microbiome, make us smart, make us more intelligent in our reactions? At least physiologically, they would make us even more alert, maybe, and cognitively more alert and more reactive and more capable. I would say the possibility is there. Yeah.
That is so interesting. That experiment that you mentioned about transplanting the microbiome to a mouse and having it be depressed just as the original host. Wow, that is so interesting. What I wanted to ask you. One of the things that I wanted to kind of have our audience kind of understand, or at least be familiarized with, is everybody's looking for like a solution specific, like just tell me what should I eat, and based on what you've been researching and a lot of data that's been collected, it seems that things can be like very much localized, right? Like, we talked about different diets, how different people from different geographic locations have different diets, and then yet they can be still healthy. In my opinion, that just adds on to the complexity of finding even maybe potential solution of like, what is the best diet? Because we have people who are vegetarians, vegans, carnivores, you have the keto diet, all these different things that are happening. How do you, people I think are at a loss right now, what should I eat? What is healthy? Other than the fact that you should eat good quality food, right? Like you want to have food that's well produced, and not, doesn't have any indigenous toxins and pollutants and things like that. Aside from that, how do you determine what you should eat? Because like it seems that there's just so many different ways of being healthy.
Yep, exactly. So we should not recommend just one way of eating, And so you need to eat for health, but also for pleasure. It's not just, I want to keep myself healthy, but I eat something, very difficult and not palatable. So, but then I think this is where microbiome research can bring a lot of science and to answer that question and to affect the daily life of millions of those, everybody.
I feel the answer is going to be somewhere customized to individuals. And I think like what you're drawing the picture for me is that given enough of data and research in the near future, there's going to be a time where the diagnostic tools will be there to kind of custom made a diet for a specific person to keep them healthy in a geographic location. And you may need a little bit of this, a little bit of that. And that's how you're going to be kind of fine tuning the this operating system that's our body.
Yeah. So right now, our research focuses on patients. And particularly patients, like right now we have a project. We know type 2 diabetes patients, when they get infected by SARS-CoV-2, and they would develop a much more severe form of COVID-19. And why? So our hypothesis is that because at the baseline, their gut microbiota is already dominated by some opportunistic pathogens. Excuse me. As endotoxin producers, hydrogen sulfide producers, endo producers, and all those opportunistic pathogens, they already overgrow at the baseline, and they already have a higher baseline level of inflammation. So when they got infected by the virus, and that makes the situation worse, and so they got higher inflammation. And when those pathogens eventually translocate from gut into their internal organs, that will lead to sepsis. So because you have a higher population of pathogenic bacteria already in your gut, that makes you vulnerable to any kind of infection, not just the SARS-CoV-2. So then what we could help them is to help them, immediately put those confirmed cases with type 2 diabetes on a high fiber supplement, so that we can reduce the population levels of pathogens and promote the potentially beneficial ones. Even the change is modest. You probably can help them, reduce the severity, delay their going to hospital. Even delay one day would be very helpful, given such a vast pandemic.
Right.
So right now, I think our strategy is to help patients with complications that almost no available medical treatment, the unmet medical needs. So when we have, because they are urgent, they are the urgent, population need to be helped. So if we can help those, and then the whole society would generally accept the new concept and the new technology, and eventually healthy people would also change their behavior. The most difficult change is change of your dietary pattern and your lifestyle. Your
habits,
and,
your behavior.
Yeah, the habits. It's if you don't have a proper, it takes a lot of efforts.
Yeah.
And it's actually easier to change patients than to change healthy people. And so we hope we can use our research on patients as example, as evidence, and then to convince the society that we need to change. But then we also need science to tell us how to change. For example, we mentioned about the fermentable fibers. Yes, in general, fibers in whole grains, they are more fermentable than fibers in vegetables. So you should take more whole grains. However, for different people, the same fiber fermentable by my gut microbiome, but may not fermentable by your gut microbiome. Because bacteria, in order to ferment, to work on one particular fiber, they need to have the enzyme to break down the fiber into small pieces, into sugars, and then they can use. Right? If they cannot do that, they cannot use that, they cannot ferment, they cannot use that. So they need, genetically, they need to have those genes encoding enzymes for them to work on that fiber.
Right.
But different bacteria have different genetic capacity to work on different fiber. So eventually, even just taking the proper combination and amount of dietary fiber would become a huge field for personalized nutrition.
Yeah, I think this is where the answer is. It's going to be customized. It's going to be individualized based on very specific tests on individuals. But for that, what's needed is what you guys are doing, which is the collection of all that data to kind of build these models, and out of which you can kind of tease out the information that's needed to create individualized either treatments or diagnostics. And that's kind of my takeaway from this conversation. Now, I know you have a meeting, so we'll have to cut short in a couple of minutes. But before I let you go, I wanted to just your own personal opinion on, because we've been talking about fermented fiber. What about fermented foods? They are typically considered very healthy, to eat things like sauerkrauts and kimchi and yogurts, and because they're supposed to have these beneficial bacteria. What is your personal opinion on that?
I would say the fermented food, yes, in general, they should be categorized as healthy food, in general. But they are healthy probably mainly not because of the bacteria there. Over history, to prevent our food from spoiling is a major thing. So then people accidentally found out that instead of make your food free from any bacteria, and that's very difficult to do, when you don't have the technology.
Refrigeration. Yeah.
But then, if you grow some beneficial, some bacteria which are not harmful to you, so then they can actually preserve the food for you. You still have the nutritional value preserved. So most of the fermented food, they are originally developed not because of the bacteria are beneficial to us. It's because they can protect our food. They actually become a way of preserving the food. But then, if you take the food, many people they say, "Oh, after I take this, I feel much better. My regularity improved." Yeah, that would be the case. But very often you cannot tell whether it is because of the bacteria there or it is because of the fiber and inside those foods. Those foods are usually high in fiber, and particularly after fermented by some bacteria, many of the fibers become more accessible to human gut bacteria. So I would not consider, unless there's evidence otherwise, I would not call the bacteria in those fermented food are necessarily beneficial bacteria. But in general, the fermented food traditionally we eat by different ethnicity, and they can be considered as healthy food.
So thank you for that perspective, because I was thinking about how you tied in together. Maybe that bacteria is not pathogenic to you. It's good bacteria because it preserves the food, but also maybe modifying the food itself so that it is accessible, just like those fermented grains that you mentioned about, which then can help the good bacteria in your gut to flourish and restore that balance. So that's very, very, very interesting. Well, Thank you so much. I really appreciate it. I wish we could talk a little bit more. I know you have a meeting, so thank you for spending this time. You definitely have highlighted some of the different perspectives that I haven't thought about in terms of the microbiome, and so I really appreciate your time over here. And I would love to invite you at some point again in the future to talk about new findings that they will have or anything that you can share. So really, thank you for being on the show. Apodcast.com. Listen, learn, and share.
Thanks for listening to A Podcast with Interesting People. If you like this episode, please subscribe and leave a review or comment.
And support us by sharing our episodes on social media. We really appreciate it. And for more information, visit us at apodcast.com. Thanks.
Thank you.